introduce our first speaker, Mr. Hanjie Wu, who will speak to us about his exciting research into the effects of microgravity breast cancer mechanisms. So,
hello, Josh. Yeah, I'm going to share my screen now.
Okay.
Wait a minute. Is it on?
Yes, we can see it.
Okay. Hello, everyone. Morning. Welcome to second Australian Space Biology Symposium. My name is Hanjie Wu. I'm a PhD student in School of Biomedical Engineering, UTS. Today, my topic is about the molecular mechanism of mechanical force uploading on breast cancer cells. So just a little bit of background about the breast cancer. So breast cancer is over 2 million new cases worldwide in 2018, and is the second leading cause of cancer death in women. So what makes breast cancer so many incidence cases is now the environmental and lifestyle factor increase the cancer cases, such as the frequent consumption of the carcinogenesis materials such as cigarettes or alcohol. So as you can see, there's so many cases worldwide each year, and this must bring lots of health and economic burden for the whole society, for the family, and the patient itself. So for normal patient, breast cancer is breast cancer, but for biologists, breast cancer, we classified it into 7 subtypes by their molecular characteristic. So as you can see that 80% of new early cases of breast cancer, they're curable actually. But metastasis, which means the cancer cells that emerge from the primary organ to other organs, is the main cause of cancer patients' death. Among of all these 7 subtypes of breast cancer, there are very special subtype, which is called triple negative breast cancer, which composed of 15% of all the cancer patient cases. It lack of the free hormone receptors, which make it no response to the traditional hormone therapy, which we are usually used in hospital nowadays. And the cancer is suffering from particularly poor prognosis. So my research, I investigate how the mechanical force are noted on this breast cancer. I'm using the triple negative breast cancer model, which called MDA MB231 here. And I investigate how the mechanical force are noted to the effect of the cytoskeleton, and then furthermore, how to affect the cancer cells' life, or even the proliferation, or they even died. So for among all the mechanical forces, the critical mechanical force is the gravity. For all the organisms on Earth, gravity is the most mechanical forces they experience, and that's why the evolution many years ago, and from how we are look like these days. But when you're talking about gravity, how to unknown gravity, the first idea coming into mind, yes, go to space. Yes, we did. We have many experiments done in the International Space Station. Astronauts up there, they observe that abnormal cell morphology when they do the cell experiments in the ISS station. And they also observe the disruption of the cytoskeleton network inside the cell. And more deep into the molecular level is the altered cell signaling and the disrupted gene expression panels. But as you can see that there's a less chance to go into space. But do we still have chance to unknown the mechanical force, especially unknown the mechanical gravity force? Yes, we can. We developed a few kind of simulating microgravity device. So one of them we are using in my lab is called random positioning machine. As you can see on the right-hand side video here, it's the two-arm rotating device. And we mount a cell culture first, which you can see in the middle of the device is rotating with the red colored cell culture media here. So this rotation random positioning machine can simulate the microgravity effect, which can down up to -3 powers by the gravitation vector aberration. We are not saying that immediately eliminate the gravity. That's not possible, but we are averaging the gravity effects on the cells. So what I've done is that I put cancer cells into the culture cancer cell into this device, and as the cancer cell the culture in this device for 24 hours, they divide into two kinds of phenotypes after just 24 hours. So they respond to the simulating microgravity really quick. So one kind of cell you can see here on the top right corner is, we call it multicellular aggregate. These are cells detached from the surface and they clump into each other to form a 3D structures, while others, they are still kind of resistant and they're still attached on the cell surface. So after that, I would like to know that if the cell forming the multiple cellular aggregate which they detach from the surface, they're still alive. So we take the culture cells out and then we put the cells back into the normal condition, like just static condition. And yes, they still alive. They still have the memory to reattach and form another monolayer on the surface, which remind me that yes, these cells are not died or just under 24 hours. We are going to have a longer term experiment, but at the time point of 24 hours, they are not 100% died. So next, we are testing the cell's migration abilities on the wood healing axis. So this is the methods that testing the cell, how the cell migrate into other locations. So cell migration is a fundamental process in the organism development, like immune response and wound healing. But for cancer cells, this is the ability for them to travel to other organs, which, just like I mentioned, the metastasis, which cancer cell emerge the shed from the primary organ into other organs, which is the main cause of the cancer patient. So as you can see from here that for the control sample, after 24 hours, they almost close the gap. The cancer cell travel from 2N towards the middle and they almost close the gap. But for the cancer cells under simulated microgravity effect, the migration efficiency is operating much slower than the control sample. So we can tell from here that yes, simulated microgravity does affect the cancer cell migration ability. And in other words, is it possible to slow down the cancer cell growing or their ability of traveling to other organs? So we have a deeper look into the molecular level. So I investigate how the vimentin looks like in the breast cancer cell. So vimentin is structured protein of intermediate filament. It's the main component of cytokeratin. So cytoskeleton is like a scaffold in the cell. It's like a scaffold for the building, and it's just like just a network for the cells. So cells need to rely depends on the cytoskeleton network to perform their normal condition and to divide into total cells. As we can see from here that after 40 hours of microgravity, the chromatin, they form from a loose network to be organized near the nuclei. And we also showing that from the protein expression level, the chromatin translation level is a significant drop after the simulated microgravity treatment. So the next one we are looking for is the trip before. It's a mechanosensation structure. It is a calcium ion channel transduction which located on the cell membranes. It transfers the iron from outside the cell to the internal cell environment. As you can see from here, that the trip before work shows differently from the previous chromatin genes. It remains stable in the normal condition environment, but it drops significantly after 24 hours culture, but it adapts to the normal expression level after 40 hours, which implies that the trip before is kind of adapted to the simulated microenvironment. After that we investigate the mechanosensitivity, we do massive parallel screening for the job target panel. Why we are doing this is because, well, we are hoping the simulated microgravity will somehow trigger the mechanical sensing pathway, and then it gives us some ideas of developing some anti-job therapy. So we identify some significant change genes after significant cancer job target genes after microgravity treatment, and then we map it back with the target gene database and try to go through that. What kind of genes, what kind of targets we are going to use. So just a head up here that we are not going to send them. You are saying that you may remember that we are using the microgravity device to rotate in the cancer cell, but we are not saying that we are sending patients into that rotation device for the treatment. What we are doing is that we are developing a supplementary treatment based on the simulated microgravity environment data, and then works with a complementary treatment with the existing cancer therapy.
1 minute.
Yeah, great. So just a summary of my topic is that I use the triple negative breast cancer cell line to put it under the simulated microgravity environment. And then we figure out that it disrupts the cancer cell from RNA or protein level, which further change is the proliferation profile and the migration abilities. So we also found that the simulated gravity informed the 3D culture cancer cell structure, which provide us a unique model for cancer job screening. So we do have some further plan, and we are future development investigate how and why the simulated gravity prevent the cancer cell from growing. We are doing more molecular work, which is due to time limiting, we didn't show here. And we are trying to identify the mechanical sense and hotspots for the cancer job development. And what's more, we are talking about simulating microgravity. And we also cooperate with institutes and governments, agents, companies to launch a cancer cell to the real microgravity environment. This is for real, not simulating microgravity. So at last, I would like to acknowledge my supervisor, Joshua Cho and Gilby here, and my lovely members in Cho Collaborative Group, and the HDR student community and academic and the School of Biomedical Engineering. Last but not least, we thank you are supported by the UTS, Mayo Clinic, Australian Space Agent, and the New South Wales government. Thank you.
Okay, thank you, Hanjie. In the interest of time, we won't have much of we don't have time for Q&A, but you can save your question for after the morning session. We'll have an opportunity for people to ask more questions. So